Hi all, today we are discussing about industrial alcohol and organic acid, citric acid, lactic acid and vinegar. Industrial alcohol. Industrial alcohol is distilled ethyl alcohol, normally of high proof, produced and sold for other than beverage purposes. It is usually distributed in the form of pure ethyl alcohol, completely denatured alcohol, specially denatured alcohol, and proprietary solvent blends. Industrial alcohol is commonly employed as a solvent and to lesser extent as a raw material for chemical synthesis. Industrial alcohol, also called ethanol, a type of industrial alcohol. Alcohols can be made through the hydration of alkenes or the reduction of aldehyde, ketone, acid, and esters, among other methods. It is used to process vaccine, compound tonic, syrups, etc. Production of industrial alcohol. Ethanol may be produced by either by chemical method or by fermentation. Next one is organic acid. Organic acid find wide use both as additives in the food industry and also as chemical feedstocks. Fermentation process plays a major role in the production of most organic acid. All of the tricarboxylic acid cycle can be produced microbiologically in high yields. Some acid derived indirectly from the Krebs cycle, such as itaconic acid, can likewise be produced. Other organic acid are derived directly from the glucose. Except for the production of citric acid, which is manufactured entirely by fermentation, there is frequently great competition between microbiological and chemical processes for production of various organic acid. For some acids, like lactic acid, acetic acid, microbiological and chemical methods of preparation are simultaneously in use. In that, first one is citric acid. Citric acid has been known as natural plant substance since the end of 19th century, and since 1893 scientists have known that it is produced by filamentous fungi. In 1923, the first practical fermentation for the production of this organic acid was started using microorganisms growing in surface culture. Fermentation using deep vat fermenters began in the 1930s, although in South America and Mexico there are some small factories which citric acid is isolated from unripe citrus fruits. Today, over 99% of the world's output of citric acid is produced microbially. Uses of citric acid, some of them are included here. Citric acid is marketed as citric acid 1-hydrate or as anhydrous citric acid. More citric acid is used in the food and beverage industries. The flavor of fruit juice extract, candy, ice cream, and mermaid is enhanced or preserved by the addition of citric acid. And some other uses are also included here. Production process. Citric acid is produced by both surface and submerged process of fermentation. Surface process can be further subdivided according to the state of culture medium used, solid or liquid. Two types of submerged processes are used: stirred fermenters or airlift fermenters. Several factors affect the choice of production type are availability of investment capital, energy availability, cost of labor and training, and availability of technique for measurement and regulation. Some factors for production of inoculum. The material used as inoculum for citric acid production is spore suspension. Spores are produced in glass bottles on solid substrate at 25 degrees Celsius with incubation time of 10 to 14 days. The detailed process is explained here. Surface process. Surface process employing solid substrate may use either wheat, bran, or pulp from sweet potato starch production as a culture medium. In this process, the Niger strains are not as sensitive to trace elements as in the other processes. And the detailed process is here. What is the sterilization period, incubation period, and the duration of fermentation, all are included, detailed in this slide. This picture, it represents the surface method of fermentation in citric acid. You can go through this. Next one is submerged process. 80% of the world's supply of citric acid is produced by submerged processes. Submerged process, although taking a longer time, has several advantages and disadvantages. All are included here. The process are detailed in this. As it said earlier, the fermentation period, incubation period, duration, etc. are all included in this. Next one is vinegar. Acetic acid is the main component of vinegar. There are some uses of vinegar. We all know that it is used as a preservatives in food. Acidity or sourness of vinegar brightens the flavor of food and adds balance to the rich dish. And some points about acetic acid are also included here. The trickling generator process is still used today. Beginning in 1949, submerged processes were developed. Both two types of process, the trickling generator and the submerged fermenter, are used worldwide. The older method still being used because of the better flavor of the product. Although acetic acid is produced by many fermentative bacteria, only members of a special group, the acetic acid bacteria, are used in commercial production. Acetic acid bacteria can be divided into two genera, gluconobacter and acetobacter. The first group oxidizing ethanol solely to acetic acid, and the second group able to oxidize ethanol first to acetic acid and then further to CO and H2O. Member of the genus Acetobacter are gram-negative and acid tolerant. Strains are commercially belong to the species Acetobacter aceti. Production of vinegar. There are two types of fermentation, surface and submerged. Starting material with low alcohol content such as wine, whey, malt or cider do not require any additional component to constitute a complete nutrient solution. However, if potato or grain spirits or technical alcohol is used, nutrients must be added in many cases to obtain optimal growth and acetic acid production. In submerged fermentation, concentration of nutrients must be 5 times higher due to the increased biomass which is taken out of the reactor with the vinegar. The inoculum can be either pure culture or vinegar from previous batches. And the surface process, as said earlier, the process has been detailed here. Surface processes. Despite the success of the submerged process, the trickling generator is still widely used in vinegar production, especially for making household vinegar. The wooden bioreactor has a total volume of up to 60 meter cube, is filled with beechwood shavings. And the whole process of surface process is explained here. And the temperature, temperature and the incubation, duration, fermentation, all are included. These are the pictures of trickling generator and submerged fermenter. Submerged process. Fruit wines and special mashes in low total concentration of alcohol were first used in submerged processes. With such low yielding process, aeration was not critical, but in present day high yielding processes which produce 13% acetic acid in quantities of up to 50 meter cube, aeration must be highly regulated. The fermenter resemble other bioreactors. Next one is lactic acid. The first microbial production of an organic acid was the production of lactic acid. Two kinds of lactic acid bacteria are recognized: heterofermentative and homofermentative. The heterofermentative organism produce a great number of byproducts and are not suitable for commercial purposes. With homofermentative organism, as little substrate as possible is converted into cell material plus byproducts, and as much as possible is metabolized into lactic acid. The biosynthesis of lactic acid from glucose proceeds by glyceraldehyde 3-phosphate 1,3-diphosphate glycerate and pyruvate. So things we all know that process. Fermentation time is about 72 hours. Since free lactic acid is toxic to the organism, procedures for removing the product continuously have been studied. In one approach, electrodialysis has been used. In other procedures, continuous culture was done in a membrane reactor, leading to the production of E. Thank you. These are all about